Hi everyone, welcome to the intervention lecture. In this session I'm going to talk about some of the different types of therapeutic intervention we can utilize against infectious disease. So this is the microbiology week of MD5027. So obviously we're more interested in infectious disease than other types of disease, and that's what we're focusing on here. And as with all the lectures, the information you're given should be informing your approach and understanding of the case study which the assessment for the week is centered on. So again, this session talking about therapeutic interventions. Now, usually when we're referring to therapeutic interventions, we're talking about interventions that will target the microorganism. We know that infectious diseases are caused by microorganisms, so the microorganism infects a host and causes disease. So primarily our approach is to clear the microorganism from the host. So we are targeting the microorganism. If the microorganism is a virus, we're using antivirals. If the microorganism is a bacteria, we're using antibiotics, antifungals for fungi, etc., etc. And this is a huge part of our approach to therapeutic intervention. However, it's not the only approach. We can also intervene on infectious disease in other ways. One example is the use of antitoxins. Now, in the microbial pathogenesis lecture, I discussed some of the different ways microorganisms cause disease, and I introduced the idea that we can intervene on disease by disrupting the ability of microorganisms to cause the disease. So we are disrupting the process of microbial pathogenesis. So this is slightly different to when we're talking about targeting the microorganism directly with antivirals or antibiotics. Well, now we're thinking about this slightly different approach, which is to target the process of microbial pathogenesis. And a really good example of this process being targeted is the use of antitoxins to neutralize exotoxins released by bacteria. We looked at some other examples as well. I think we've discussed the use of particular drug-based interventions to disrupt immune activation by microorganisms. So some microorganisms will cause disease by inducing immune function. And if we can disrupt this process, so we can intervene and prevent particular elements of the immune system from being activated, we can prevent the microorganism from causing disease. So far, we've kind of focused here on interventions on active infections. So what can we do to target an infectious case? However, when we're talking about interventions, we can be more rounded in our understanding, and we can also talk about interventions before infection occurs. So we can intervene before individuals contract an infectious disease, and we can prevent them from contracting it, or at least reduce the likelihood of them contracting it. And this approach is considered prevention, it's considered prophylactic. You're doing things before the infection occurs to prevent the infection from occurring. And obviously the prime example of this is something you're very familiar with. It is vaccination. We vaccinate individuals, generally speaking, before they are exposed to an infectious agent. And in doing so, we are preparing their immune system, we are fortifying their immune system, we are making them resistant to infection by a particular infectious agent. There's kind of qualifications here. So vaccination isn't always before you're exposed to an infectious agent. Sometimes once you're exposed, you can then become vaccinated, like with rabies. However, you are at least becoming vaccinated before the infection takes hold. So the idea is you're preventing the full infection from taking place. And these are some of the things we're going to talk about today. So then that was just a bit of context about the overarching themes, what we mean by intervention, the different types of interventions. Obviously, we don't have the time to go into every single type of intervention in this session. However, what we can do is look in some detail at the examples I've just introduced. So we can look at antibiotics in a bit of detail, we can look at antiviral drugs in a little bit of detail, and then we can look at vaccination in some detail as well. And hopefully by doing so, it will give you a good understanding of the principles of intervention. So antibiotics are interventions that target bacteria specifically. So what they're doing is they are targeting the bacteria that are causing the infection. Now they can work in a range of different ways. There's different types of antibiotics, but what these antibiotics have in common is they are disrupting the bacteria. Okay, they are targeting the bacteria, they are either killing it or they are preventing it from growing, and in doing so they are hopefully clearing infection. Um, as we said, various types of antibiotics, and they're generally categorized by the type of bacterial process they inhibit. So we have the bacterial processes on the left here: cell wall synthesis, protein synthesis, nucleic acid synthesis, metabolic pathways, and cell membrane function. So these are 5 processes that are clearly important to bacterial survival. So it is from our understanding of both microbiology and cell biology, it should be pretty clear to us that if you really disrupt any one of these processes, that is going to cause significant problems for the bacterial cell in terms of survival. How can a bacterial cell survive and replicate if it is not synthesizing proteins, if it is not synthesizing a cell wall, if it has disrupted metabolic function? And this is the principle. We are taking a drug-based intervention and we are disrupting these bacterial processes. Now, there's a central theme running through this idea of intervention, and the theme is that there is a need to avoid toxicity and side effects. So bacteria are prokaryotic, so a bacterial cell is a prokaryotic cell, whereas our cells are eukaryotic. So when we're using drug-based interventions, we want to specifically target bacterial cells and not target host cells, because if we're targeting host cells, we have toxicity, we have side effects, and this is obviously something we really don't want, because if a drug is associated with side effects, then it's not useful, is it? It's dangerous. If the side effects are severe enough, then it can't be taken. So what these antibiotics do is they tend to target bacterial processes specifically. So they will either target processes that only occur in bacterial cells, so they don't occur in our cells, or they will target processes that occur in both cells. However, they will only be targeting the process in the bacterial cell because they target it in a specific way. So for example, protein synthesis, this is a process that occurs in bacterial cells and host cells. However, antibiotics will only target protein synthesis in the bacterial cells because bacterial cells conduct the process in a slightly different way to the way in which eukaryotic cells conduct it. And by this mechanism, by this strategy, antibiotics are being utilized to specifically target bacterial cells rather than eukaryotic cells, and therefore side effects are minimized. This is very clearly demonstrated when we consider peptidoglycan. So peptidoglycan is a core component of the bacterial cell wall. It's only present in bacteria, it's not present in eukaryotic cells. So when we have developed antibiotics to specifically target cell wall synthesis, so specifically target this process of peptidoglycan being built up and string together to make the bacterial cell wall, we are specifically targeting bacterial cells, and therefore we're avoiding toxicity. And there's some really important groups of antibiotics, including beta-lactams, that target peptidoglycan, so they target the synthesis of the bacterial cell wall. The specific mechanism is on the right. Again, we don't need to know this. It's there for you to have a look at if you want to have a look at it, and we'll cover it in more detail next year. It's covered in 6-year module, level 6 modules. But the main thing we want to understand is the principle. We're talking about antibiotics that are specifically targeting bacterial processes, or are targeting processes that occur in bacterial and eukaryotic cells. However, they are targeting a unique aspect of the process that is only occurring in prokaryotic cells. So like the example with protein synthesis. And this is what we have on this slide. So as we've said, protein synthesis occurs in bacterial cells and eukaryotic cells. However, it occurs in a different way. So the antibiotics that target protein synthesis specifically target protein synthesis in bacterial cells. And this is possible because the process is slightly different. There's slightly different enzymes used. The process overall is slightly different stage by stage, and therefore we have managed to we are able to use antibiotics that specifically target protein synthesis in bacterial cells and not in eukaryotic cells. We also have inhibitors of nucleic acid synthesis. Again, as you're seeing, we're talking about really core fundamental cellular processes: protein synthesis, cell wall synthesis, nucleic acid synthesis. This is why antibiotics are so effective at disrupting bacterial reproduction and or survival, because they are inhibiting key, important, crucial processes within the bacteria. Now, as we said, nucleic acid synthesis, it's like protein synthesis. It's occurring in bacterial cells and eukaryotic cells. However, the antibiotics will preferentially target the process in bacterial cells. Okay, so they will preferentially be having an impact on bacterial cells rather than eukaryotic cells. We have two more groups here. We have the inhibitors of metabolic pathways, and again we have this difference between bacterial cells and eukaryotic cells. So when we're talking about the metabolic pathways of bacteria, particularly those involved in nucleic acid synthesis, so those upstream, there is something evolved called hydrohydrofolic acid, THFA, and this is utilized in the pathway that bacteria follow, but not utilized in the pathway that eukaryotic cells follow. Okay, so antibiotics that specifically target the process involving THFA will specifically target bacterial metabolic pathways, but will not specifically target eukaryotic metabolic pathways. Finally, we have inhibitors of membrane function. So this is, again, we're talking about membranes. We're talking about something that could impact bacterial cells as well as eukaryotic cells. However, the principle is the same as usual. The differences between eukaryotic and prokaryotic cells are sufficient that antibiotics can target bacteria without really causing harm to eukaryotic cells. The issue with inhibitors of membrane function is the principle doesn't play out very well, and we actually see these drugs do have toxic effects. They do have toxicity. So the differences between the membrane function are just perhaps not quite sufficient. So these aren't very widely used anymore. Next, we want to discuss antitoxins. Now, antitoxins, as we've mentioned in previous lectures, are interventions that disrupt the bacterial exotoxins. So in the microbial pathogenesis lecture, we discussed how bacteria, or many bacteria, release exotoxins into the local environment, and these exotoxins cause disease. Now, one strategy to limit the ability of these bacteria to cause disease is to neutralize those exotoxins, and this is what antitoxins do. So antitoxins are administered to a patient and they neutralize the exotoxins that are present in the system, and this is done in a specific way, of course, and there's specific strategies to develop and obtain the antitoxins that are used in various situations, and this is what's outlined on the right. So this is the strategy for isolating antitoxin to neutralize diphtheria toxin. So we talk about Crinibacterium diphtheria being one of the bacteria that release a specific toxin and that causes disease in the host. And again, very different situation with antitoxins to antibiotics. So antibiotics are used to target the bacteria. This is why in the last few slides we were talking about the different types of antibiotics and the specific bacterial processes that they target, because antibiotics are targeting bacterial processes. They're disrupting the bacteria. This is not what's happening with the antitoxin. Antitoxins are not targeting the bacteria itself. They're neutralizing the toxins that are released by the bacteria. This doesn't necessarily mean they're less than the antibiotics. They're less useful. They're less effective, because the exotoxins released by some bacteria can be really, really powerful. they can cause really quite severe disease. And so the use of an effective antitoxin can be very, very useful therapeutically. Next, we want to discuss antiviral drugs. And again, I'm really not concerned about you memorizing any of this information. I just want you to understand it. Um, it's it's quite unfortunate that even at level 6 and even occasionally at master's level, unfortunately, there'll be misunderstandings with students about antivirals and antitoxins and other symptomatic interventions and antibiotics, and there's sometimes misunderstandings about how they work, what they're intended to do, how they function. So we're really just trying to clear up any misconceptions, make sure everyone is really clear about this as they're coming out of this module and heading towards level 6. Now, antivirals, they're not antibiotics, they're not working in the same way as antibiotics, but they are working in a similar way, in a comparable way, against viruses, whereas antibiotics are working against bacteria. So the antivirals and antibiotics targeting different processes, they're targeting different microorganisms, but they're doing so in a comparable way. So what antibiotics are to bacteria is what antivirals are to viruses. The way antivirals target viruses is different to the way antibiotics target bacteria, and this is necessary because viruses and bacteria are completely different. Because they're so different, the approach that our therapeutic interventions take has to be different. So we develop therapeutic interventions that work in a different way because we are dealing with different groups of microorganisms. We know that bacteria are cellular microorganisms, and therefore the antibiotics we have developed and use, target cellular processes. Protein synthesis, nucleic acid synthesis, processes to do with the cell wall and cell membrane, and they work very well. Antivirals, on the other hand, these are targeting viruses, and we know viruses are not cellular. They don't have a cell wall, they don't have a cell membrane, they don't synthesize protein using their own machinery. They actually use cellular machinery, so it's a different approach we have to take. We can't target the same processes that we target in bacteria. Now, it's actually really difficult to develop antivirals, and if you did my module last year, you remember, or hopefully you remember, we talked about this. We talked about the idea that when we're developing antivirals, it's really, really difficult because viruses use host cell processes. So viruses enter into a cell, they manipulate that cell, and they utilize the host cell machinery, they co-opt that machinery. So it's very difficult for us to disrupt that process without also disrupting host cells. So without producing something that is very, very toxic. Because again, the virus is using the host cell processes, it's using the host cell machinery. So we can't target that without also targeting the same process in healthy cells, right? Very, very difficult. I shouldn't say we can't do it, I should say it's very, very difficult to do it, because obviously we have managed to do it in some cases, and we do have some effective antivirals, not loads, around 100, maybe a bit more now. But again, it's very, very challenging, and we're constantly trying to develop new antivirals. There's other specific challenges as well. It's very difficult to work with viruses, they're very dangerous. We often don't have the correct animal models we need to test these antivirals. So a lot of very, very specific challenges and very different challenges to the challenges that we're faced with antibiotics. So with antibiotics, one of the main challenges is antimicrobial resistance. The idea that bacteria will develop resistance to these antibiotics. You do have an issue of antiviral resistance, but overall the issues with antivirals are broader, and there's a lot more and a lot very different issues. Now, it's interesting that of the antivirals we've developed, most of them have been successfully developed against HIV, though a lot of them also work against other chronic viral infections. These antivirals, almost all, work in a similar way to each other in the sense that they target the viral replication cycle. So the successful antiviral drugs successfully disrupt this viral replication cycle. You should know what the viral replication cycle is. You should understand the principle that viruses are obligate intracellular parasites. So they enter into a host cell where they replicate and they make copies, and these copies then leave the cell and continue infection. If you don't understand these processes, just make sure you've read up on them, because this is something you should have covered at level 4. And again, as we should know, the viral replication cycle is split up into distinct steps. So there's 6 different steps, and antiviral drugs are disrupting these 6 steps. For some examples, here we have merivaroc and enfuratide. These are drugs that are inhibiting the attachment of the virus to the cell surface and the entry of the virus into the cell. Again, if you don't know these steps, you really just need to go away and read them. They're in the textbooks. I'd encourage you to read up on them. The principle, however, is that there are these specific steps that the virus engages in as part of its life cycle. These are not cellular processes in the same way that bacteria are undergoing their own specific cellular processes. With viruses, these are just ways that the virus interacts with the host cell. These are ways that the virus engages in its obligate intracellular life cycle. And the start of that life cycle is the virus attaching to the surface of the cell and then entering into the cell. And with antivirals, we can disrupt these processes, and therefore we can prevent infection. The next 2 stages, genome replication and genome expression. So this is where the virus replicates its genome and expresses its genome. This occurs inside the cell, and again, if we can disrupt these processes, we can prevent the viral life cycle from taking place. So we can prevent the virus from making copies of itself and continuing infection. Acyclovir is a drug that successfully does this. It's utilized against a range of different herpes viruses infections, and again, the principle is it's targeting viral genome replication and viral genome expression. Now, the question arises, how does acyclovir manage to specifically target viral replication? This process is going on in a host cell. How does acyclovir not target host genome replication and host genome expression? Well, the answer is that acyclovir only works when it is processed by a particular viral enzyme. So it only works when it's processed by viral thymidine kinase. So when it is processed by this thymidine kinase, it turns into its active form, and then it can inhibit genome replication and genome expression. Okay, so therefore it needs to be activated, and when it can, it inhibits these genomic processes. It essentially competitively inhibits the process, so it competes with the normal cellular process going on and reduces genome expression and replication. Now, if the viral thymidine kinase isn't there, this process never happens because acyclovir isn't processed to its active form. And this is one of the ways that researchers have very cleverly got around this idea of toxicity. So for researchers to get around this problem of viruses using host cell machinery, they need to approach it in some very interesting and unique ways. And this is one of the ways that is quite ingenious. And the drug only becomes active when it is activated by the viral enzyme, and the viral enzyme is thymidine kinase. Finally, we have the stages of assembly and release. So the new virus particles, after the genome has been replicated and expressed, must assemble. So the viral genome copies are surrounded by viral protein. Again, if this is not sounding familiar, you need to read up on this. And then these new viral particles are then released from the cell. Now, some antiviral drugs will prevent this process of release, so they will prevent the virus particles from exiting the cell. Some will also prevent the process of assembly, so as the new viral proteins are being chopped and changed and they're being put together around the viral genome, there are specific processes that involve often specific viral enzymes. So some viruses carry their own protease, and this protease is an enzyme that cuts up the viral proteins in a specific way so they can fit together So it's not a host cell protein, it's a viral protein, it's a viral enzyme. Um, and some drugs will target the viral protease. And again, this is another way of avoiding toxicity. Viruses have a small number of their own enzymes, and many antivirals will target these viral enzymes specifically, again, to make sure that the antivirals don't impact healthy cells, that they're specifically targeting viral processes. Okay, so far we've gone through some of the different therapeutic interventions that we can use to treat established infections. We can also think about vaccination as being the prime example of a preventative intervention. So this is an intervention, it's something that can be done to prevent infection from occurring. So the idea is this is an invention we apply to healthy individuals to reduce the likelihood that they contract a particular infectious disease. Now, the details in terms of immunological details of how vaccination works, beyond the context of what we want to talk about now. However, we do need to understand what we mean by vaccination. What we essentially mean is we are priming an individual's immune system. We are priming the individual's immune system, which makes them immunologically prepared for an encounter with a particular infectious agent. And when they are immunologically prepared, they are immunologically incompatible. So they are not compatible with the microorganism. The microorganism can't establish infection with them because their immune system is able to fight off the infectious agent. And this is what we're trying to achieve with vaccination. Now, vaccination is something that we can evaluate in a really, really interesting way. A little bit beyond the scope of what we're talking about here, but I've put some interesting evaluative points on the slide. Now we've moved from level 4, we're right at the end of level 5 and we're moving into level 6. We should be really familiar with evaluation at this point. If we're not, this is a good example of how to evaluate. We take something, whether it's a diagnostic test or it's a therapeutic intervention, and then we are able to discuss the benefits of this and also the negative sides of this in an objective, reasonable, rational way. So what are the strengths, what are the weaknesses, in what particular circumstances should this be applied and in what circumstances should it not be applied. Every single intervention, every single diagnostic test has strengths and weaknesses, and we want to get better and better at understanding these and interpreting them. Okay, here we have the idea of herd immunity. We can't really discuss vaccination without discussing herd immunity, and the idea is that firstly, if an individual is vaccinated, they gain an individual protection because they are immunologically incompatible with the infectious agent. But also, if we have enough individuals in a population with this individual protection, they confer a level of protection to other individuals in the population who haven't been vaccinated. So if we take an individual who hasn't been vaccinated and we say this individual is surrounded by individuals who are protected, so who have been vaccinated, then the individual in question who hasn't been vaccinated, they will obtain a kind of de facto protection because they will never come into contact with the infectious agent because they are surrounded by protected individuals. So even though they are immunologically compatible with the infectious agent, they obtain a layer of protection, they obtain a degree of protection by being surrounded by individuals who are not compatible with the infectious agent, who cannot carry the infectious agent. On the slide here, I've got a graphic demonstrating this fact. It's not going to play through the video. However, you can download the PowerPoint slide from the module space, MD5027, and you'll be able to see this play out. Essentially, there's 6 different populations, and in each of these 6 different populations, there is a different level of vaccine coverage. And as you'll see, or as you'll see when you play this graphic, the higher the level of vaccine coverage, the greater the level of protection for individuals who are not vaccinated. And this is referred to as herd immunity. There's different types of vaccines. Again, this plays into the idea of evaluation because the different types of vaccines have different strengths and different weaknesses. And if we want to understand in detail and really discuss how these vaccines can be used, we really need to understand the strengths and weaknesses. So if this is something you're interested in, I'd encourage you to have a read up on these types. We have live vaccines, killed vaccines, and attenuated vaccines. I won't go into them now. I don't think it's particularly relevant for us now, but the information is there if you want to look at it in more detail. Okay, everyone, thank you for your time. That's the end of the session. Again, hopefully it's been useful. It's been a walk through the different types of therapeutic interventions we can use. Hope it's opened your eyes a little bit to the different types of therapeutic interventions, as well as the different strategies we can use when developing therapeutic interventions. So largely when we're talking about therapeutic interventions, we're talking about targeting individuals who carry an infectious agent. However, not always. We can also think about prevention strategies under this banner. Okay, so thanks very much for your time, and I'll speak to everyone soon.